Good morning, children. Today we are going to discuss molecular characterization of eukaryotic gene. So, eukaryotic gene means what? U means to everybody know karyotic gene where the nucleus is present, right? So, molecular level we are going to discuss and molecular level how the genes are arranged. How the genes are arranged in eukaryotes. So, here when you take the fundamental unit of life that we call them as a cell, right? In this cell we have the two different types of chromatin materials which are hidden inside. One we call them as euchromatin and another one we call them as heterochromatin. Heterochromatin, right? So in euchromatin we found actively participated chromatin material that we call them as chromosomes. Now, in heterochromatin they are inactive, but sometime due to the facilitative mutations or any type of mutations, these heterochromatins are going to convert it into active form and they show some sort of disorders. So, we are discussing regarding how the molecular organization of eukaryotic gene based on euchromatin. Now, we will concentrate euchromatin material. Euchromatin material where we have the 22 autosomes, only one pair of sexual chromosome XY and XX. XY in case of male we found and XX in case of females we found it. Now we are focusing especially the genes which are arranged in them, right? Now when you open the chromosome out, we have the 22 chromosomes which are autosomal in nature, and we have one pair of sexual chromosome XY in case of male and XX in case of females. Now, in chromosomes, the thread-like arrangement we call them as chromonimeta, where the DNAs are tightly bounded around it in the form of nucleosomal complex. These are all nucleosomal complex which are a bead-shaped structure which tightly coil the DNA in presence of basic histone proteins. Whenever you observe this single bead, and it was studied by Corona, an Indian born American scientist who worked on bead structures along with Korenberg. Now, to study how the arrangement of basic histone protein around the nucleosome. Now we call them as nucleosome. A bead-like structure we call them as nucleosome. In this nucleosome, how the basic histone proteins are arranged. It are arranged in the form of a bead-like structure. We call them H2A proteins, H2BA proteins, H4, H5 proteins. When we write here, this one is H2A, H2B, H4, H5, H4, H5, H2B, H2A. What these are? These are all basic histone proteins. What they are? Basic histone proteins. And these basic histone proteins are those, they have what charge beta? They have positively charged. As we know that the DNA is having the negatively charged, so this DNA which is having the negatively charged, they combine with the positively charged of basic histone protein to form a tightly coiled. That means positively charged basic histone protein combined with the negatively charged DNA. In this DNA, phosphoric acid is there, H3PO4. To the presence of this negatively charged phosphoric acid of the DNA and due to the presence of positive charge of basic histone protein, they two get attract with each other and they form tightly coiled around themselves, so that the DNA is tightly arranged around the basic histone protein complex. Another interest fact here in terms of how octameric complex it is known as. The nucleosome is known as octameric complex. Octameric complex. Why it is known as octameric complex? Because when you count here, we have 1, 2, 3, 4, 5, 6, 7, and 8. The whole complex we call them as octameric complex. Now, so here one interesting fact here is these basic histone proteins are made up of basic amino acids. Basic amino acids. What are the basic amino acids? In an entrance point of view or any common entrance in life science examination, we found what are the basic amino acids which are responsible for the formation of basic histone proteins. Means lysine and arginine. Important, Nana. Right. So far we have learned about how this eukaryotic gene is hidden inside the euchromatin material. Now we will go with in deep details about how the eukaryotic gene are present in given chromosomal autosomal chromosome. Right now, when you cut any part of this chromosome, this part we call them as DNA. Now we will learn when you open this stretch of this DNA how the genes are arranged. We will learn now the next upcoming event about the DNA. In this DNA, how the genes are arranged. Can I rub the board? Yeah. So you people, you are aware about what is euchromatin and heterochromatin. In case here in euchromatin, it is fundamental where they involve in all cellular metabolism. 22 autosomes are responsible in body makeup. That means metabolic activity as well. They involve in the physical maintenance of our body. Then we have the XX chromosome and XY chromosome. They involve in secondary sexual characteristic feature. Right now, when you open out the DNA, if I cut this part to explain you, and this is a DNA. In this DNA, the genes, again I am repeating, when I open a part of this chromosome, we call them as DNA. A piece of chromosome, we call them as DNA. In this DNA, the genes, the blue color lines indicates the genes which are arranged linearly. So the position of these gene are known as locus or loci, singular and plural. Now, we are focusing on especially eukaryotic gene structure, right? Now, there is a question arises, how many genes are there in a single fragment of DNA? Means more than 100 we have the genes, and each genes involves in their gene expression individually, not an isolated way, right? We can comparative between the prokaryotic bacteria, the group of genes involved to perform the gene expression. Here it is not like that. Now, so the one gene, when you isolate it out to explain, it has a 5' N, 3' N. So central located where the transcriptional start point begins. Transcriptional start point begins. We call them as transcriptional point zone. From here, we indicated with the sign, positive sign, until the end. Before the transcriptional start point, we indicated with a negative sign. So this is the transcriptional. That means eukaryotic gene is divided into two compartments. The first compartment we call them as upstream element. It is also known as controller gene sequence. controller gene sequence. Whereas after the transcriptional start point, it is indicated with a sign by the name downstream process. Downstream element or process. Right. It is also known as structural gene sequence. Structural gene sequence. Right now. So again, when you isolated the chromosomal out, from this chromosome, if you cut any part of the chromosome, we call them as a DNA. In this DNA, the genes are linearly arranged. So when you isolate the one gene, and this gene, which is a nuclear based gene, why? Because in eukaryotes we found the nucleus, so we call them as a nuclear gene. When you compare the prokaryotic organism, in the prokaryotes we found a bacteria, and thus bacteria, there is no distinct nucleus. So the nucleoid, that means naked DNA is there. So in bacteria, the type of genes we call them as nucleoid gene. We call them as nucleoid gene right now. So this eukaryotic gene is divided into two compartments based on the transcriptional factor proteins. So it is divided into upstream element, and it is also known as a controller gene sequence. Whereas after the transcriptional start point, we call them as a downstream element. It is also known as structural gene sequence right now. Minus, see carefully, a stretch of DNA is nothing but it has having the number of base pairs among them. So minus 25 base pair away, minus 40 to minus 180 base pair away. It is having the specialized sequence, we call them as CAAT cat, TAAT tata. That means it has the two boxes, we call them as kata box and tata box. Important data. So kata box and tata box. And these two boxes are acting like a promoter. These two boxes are acting like what? Promoter. So these two boxes are acting like promoter. Right. What is promoter means? The promoter is a specialized structure which are responsible for binding of RNA polymerase. This enzyme is responsible to bind on promoter and enhances the frequency of transcription. We will discuss what is enhancement of frequency of transcription. Then next, here we have silencing gene elements. Here we have silencing gene element. This is a specialized element. We call them as gene silencing element. So these three, kata box, tata box, get together. They are known as a promoter. Where they are responsible for binding of RNA polymerase enzyme on promoter complex. Whereas silencing gene element, they helpful to control before the transcription begins. So that is what we call them as controlling gene sequence. Whereas after the transcriptional start point, here it has a leader site. Leader site is there, then followed by exon, followed by intron. Exon, intron, exon, intron. At the end of 3' end, it has a sequence by the name adenine, 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 adenine. So it is known as polyadenyl tail or end. Polyadenyl tail or end. Now, And what these two elements, what we call them as leader site, means what? Whenever RNA polymerase enzyme come in contact on the promoter, they are able to be increases the transcription level. That means from this DNA, they are going to be produced mRNA. So that structural gene is going to be expressed in gene expression. So here, exons are those they code for specific amino acid. Exons are those they code amino acids. They code for what? Amino acids. They code for amino acids. Introns are there, and these introns never code any amino acid. They never code absence of amino acid coding. Amino acid, they never code. That is a one fundamental right now. So exons are known as a coding elements. Introns are non-coding elements. These exons and introns are equally. That means number of exons are equal to number of introns. That means exons are equal to number of introns. So that is the one thing beta. In a chromosome, how many number of introns are there? If exons are 25, that means 25 equals to exon and 25 equals to intron. So that is equivalent amount, proportions they are present here right now. Now, so these are overall view related with eukaryotic gene, eukaryotic functional gene. When we enter inside, the brief discussion how they involves in their expression. Now we will go with that one, right? Again, I will briefly explain you. Eukaryotic gene, when you isolated the chromosome, in this chromosome, a part of chromosome we call them as a DNA. In this DNA, the genes which are linearly present. So position of this chromosome we call them as locus. When you isolate this locus, when you isolate the gene from this locus, when you stretch it, we have the 5' to 3' end of this gene. In this gene, what happened? It is divided into two compartments. We call them as controller gene sequence, structural gene sequence. Where the transcriptional start point begins, the gene is categorized into two compartments. One is upstream and another one is downstream. Minus 25 base pairs. As now, just now we indicate that where the transcriptional start point begins, before the site we call indicated with negative sign, whereas after the transcriptional start point we indicated with a positive sign. So minus 25 base pairs means away the base pair nucleotide. For example, 1, 2, 3, 4, 5, 6, 7, 8, 9 base pairs are there. The stretch of line is nothing but it is arrangement of nucleotide right now. So minus 25 base pair and minus 40 base pair, we have a specific sequence by the name cat box and tata box. These two boxes get together, act like a promoter, and this promoter complex is helpful for the binding of RNA polymerase enzyme to enhance the frequency of transcription. So here we have silencing gene elements. What they do means they control whenever RNA polymerase enzyme is bounding on the promoter, they helpful for the transcription process. So that's why we know as controller gene sequence and upstream elements. Now, at the point of transcriptional start point, we have the leader side to lead the transcription. So what are the elements are present in structural gene means here we have exon, intron. Number of exons are equal to number of introns. So exons are coding sequences, they code for specific amino acid. Introns are non-coding sequence, but they're helpful in forensic science where it has a variable number tandem repeats for identification of DNA profile between one individual with another individual. So in that cases, introns are playing a pivotal role in identification of mismatched babies in hospitals, murderers in crime, and rape cases. In that cases, we require this intron for identification of threat or the suspected individuals. So this is a brief structural gene concept. When we enter in detail how the expression involves, yeah, right. Now we will discuss how the expression involves in eukaryotic gene. As we know, you people are aware about gene expression. Gene expression, or by the name central dogma, right? Now, we will discuss what is a central dogma and how the expressions involve. In each gene which are present in a cell, they undergo central dogma. Example, the first gene to the 100th gene, each gene undergo central dogma. Central dogma means what? Central dogma means the flow of genetic information. Every gene should undergo their expression, that we call gene expression. Gene expression. Just now we saw how the genes are arranged in the DNA, right? Now, as you know, gene expression means what? Gene expression means it regulates the expression of the protein via RNA and DNA. Right now, we will go for the discussion. The flow of genetic information. The flow of genetic information from DNA to mRNA to a protein is nothing but gene expression. And this flow we call them as a central dogma. And this process and phenomenon we call them as central dogma. So this gene expression in all eukaryotic organism is unidirection. That means the flow is in unidirection, right? So we call them as unidirection. Unidirection. But we found such type of direction is bidirectional. Where we found in bidirectional situation means whenever the patient who are infected with HIV virus, in that virus what happened? From mRNA to again the DNA will going to be produced, we call them as, the phenomenon we call them as reverse transcription. What we call reverse transcription. Why this happened like that means in HIV virus, human immunovirus, there is a enzyme by the name reverse transcriptase. Reverse transcriptase. Due to the presence of these enzymes, they involve in reverse direction. That means from mRNA they are going to give rise to the DNA. So this concept was explained, and this concept was studied by the great scientist by the name Temenism. The great scientist by the name Temen was a scientist. So on basing on the scientist's name, it is known as Temenism. So this is a great concept related with the gene expression. Gene expression, the flow of genetic information from the DNA to mRNA to protein. Now here, the phenomenon is known as a central dogma, but here, the flow of these expression in case of higher eukaryotes like you and me, in our plants as in higher animals, we found unidirection. The flow is unidirection. But here, whenever any patient was suffering with HIV, in that HIV virus, the HIV have the specific enzyme by the name reverse transcriptase enzyme, and this enzyme will reverse back, turn back RNA into DNA. That means they are undergoing the construction of these RNA in a reverse direction by the name reverse transcription. So in that patients who suffered with HIV, the proper expression of protein will not take place. He become weak and weak. Proper cell metabolism is not seen in that particular individual who was suffering with HIV. Then slowly the body gets weakened. In due course of time, he is ready for the deathbed. So this is a concept related with structure of gene and how they involve in gene expression. We have in upcoming classes, we will discuss about gene expression, that is eukaryotic gene expression, that is a transcription we will discuss, translation we will discuss, and so far we will go with all the different types of expression in especially in eukaryotic organism. Then we will go back with prokaryotic organism, how the genes are arranged in prokaryotic gene and how they involve in their expression, why they are known as nucleoid gene and why this one is known as a nucleus gene. And this phenomenon we will discuss in upcoming classes. Thank you for listening the class. Please give your feedback so that you can give me as a feedback so that I can encourage myself to do such type of videos again and again. Thank you and all.